Welcome. Today we're going to be looking at the accessory structures of the integumentary system, or things that complement the skin. Reference pages are going to be between 176 and 180. So things that complement the skin. There are three accessory structures of the integumentary system, namely the hair follicles, nails, and skin glands. Over to the right over here we see a picture of a fingernail, and we can see a picture of the integument, which is the skin consisting of the epidermis and dermis with the hypodermis beneath it. We can see a second accessory structure right here. This is a gland. And then we can see hair right here extending from the epidermis down into the dermis. Now, while this isn't necessarily the case for the glands, it is especially true of hair and of the fingernails. They originate from the epidermis, and by that what I mean is there's a little red section down here, and then we can see these granular looking things, which are actually cells. They go around the hair down into the dermis. By originate, I mean that these cells create our fingernails or our hair. So our hair and our fingernails were once cells. They came from these cells that are part of the epidermis. The first one we're going to be talking about is our hair follicles. So some general information about hair. To start with, you have actively growing hair. Not that it's alive, but it is increasing in length, therefore it's growing. We have actively growing hair for 2 to 6 years. So one hair will grow and grow and grow from 2 to 6 years before it eventually stops growing. Once it stops growing, it will sit for 2 to 3 months before it inevitably falls out. When a hair falls out of the hair follicle, which is this entire region right around here, a new one will start to grow in its place to, replace it. If that does not occur, you have what is called baldness. So some people, they try to attempt to slow down or stop the balding process, which happens older in age. And some things that can be done to prevent that would be Rogaine, hair transplants, or thinning hair supplements. Rogaine works well at preserving hair, meaning it slows down the rate at which it falls out. So if it's supposed to be in the hair follicle for 2 to 6 years plus 2 to 3 months, it might make it 6 to 9 years or 6 to 10 years. It holds on to the hair a little bit longer than the average expectant range. On very few occasions, Rogaine will actually cause a vacated hair follicle, which was what we see on our screen if the hair were removed and nothing were filling the hair follicle. At a very minimal rate, though. Only about 10% of people report that Rogaine will cause new hair to grow from previously bald areas. There's also hair transplants, taking hair from one part of the body, plugging it into a vacated hair follicle. That works pretty well. And then there's also thinning hair supplements, which work only really when your hair is thinning. What it does is it's a condition that you put in your hair in the shower, and it will cause the hair to look thicker to fill in some gaps. At any given moment, hair spends most of its time in the growth phase. Roughly 90% of the life expectancy of hair is in the growth phase, and we do lose between 20 and 100 of these things every day, which you can probably get a pretty good sense of if you look at the drain after you get out of the shower. Types of hair varies as well. We have some really, really thick, coarse hair and some peach fuzzy hair. Thick, coarse hair is terminal hair, whereas the peach fuzzy hair is vellus hair. On an interesting side note, looking at this guy's chest, and this peach fuzz actually, we are quite similar in chimpanzees in the amount of hair that we have on our body. Chimpanzees don't really have a whole lot of this vellus hair, though, whereas we have an extensive amount over our bodies. Chimpanzees tend to have more of the terminal hair. So even though they look a lot hairier to us, we actually have about the same amount of hair. So looking at this slide here, which you have in your handout, I'm going to touch on a few things on this one, but I'm really going to be discussing these on the next slide, which shows you a picture of a lot of it. So while I'm talking about that picture, I'd like you to kind of look at this as we go through it. We're going to be talking about the hair follicle, the hair root, dermal blood vessels, the hair papilla, as well as the hair shaft. So in essence, all these things, we're going to be talking about how hair is actually growing. So to start with, let's look at the hair follicle, which is found on the screen right here. It's pointing to this very thin tan region that envelopes the entire hair, going all the way around it. That is the hair follicle. Hair is found inside of the hair follicle. When baldness occurs, the hair follicle is still there, but there is no hair inside of it. We also saw the root sheath. The root sheath, a sheath is something that envelopes something. If you were to hold a sword on your belt, you probably would put it in a case of some sort. The case that holds a sword is called a sheath, and hair is no different. It's something that wraps around the hair. We can see the outer root sheath out here, and it would wrap around the entire hair. So that's a root sheath. Looking down lower at the bottom of the hair, which is deep in the dermis, we can see the hair papilla, which is this region right here. It's indented a little bit, and what it does is it accepts blood vessels. We can see a dermal blood supply right here going into the hair papilla. The blood supply coming through the hair papilla provides the epidermal cells in this region some nourishment. What these cells will do is they'll go through mitosis. They'll reproduce new cells and push older cells upwards. As the older cells get pushed upwards and upwards and upwards, they begin to die and they begin to keratinize. Similar to what we spoke of when we were looking at stratified squamous cells. The bottom layers are alive, they reproduce, and as they reproduce, they push cells upwards that keratinize and die. Well, in our hair, the keratinized dead cells from these epidermal cells actually form the hair that we see once it reaches the surface of our skin. So this exposed hair, which is called the hair shaft, is nothing more than previously living cells. They're now dead at this point. So on your previously obtained integument coloring sheet you received, we're going to be coloring and labeling these sections of that sheet, and only these sections, because these are the ones that we just spoke of. So please label them, and then pause this video so you can take care of that. Next, we're going to be talking about hair color. Hair color, like skin color, is dictated by a pigment known as melanin. And it's found in our genes, is controlled by genes. Genes dictate which type of melanin an individual has in regards to their hair color. It's either going to be eumelanin or pheomelanin. Individuals that are really dark in hair color, namely brown through black, they're going to have a melanin called eumelanin. So what ultimately causes one person to be maybe a light brown versus a dark brown? The amount of that melanin present. So a person who's really dark black hair, they have eumelanin, and it's produced in heavy quantities. In contrast, there's pheomelanin. Pheomelanin will produce blonde to reddish hair. So it operates in the same principle. Individuals who are really light blonde have pheomelanin, but it's only produced in little small quantities. Right here, maybe pheomelanin in medial quantities. And over here, when we start to get to the brownish end of the spectrum, we might have pheomelanin produced in high volumes. If we had an individual with no hair color whatsoever, they're an albino, they have no melanin at all, the genes produce 0 melanin. You do have this chart in your handout, and although it's not nearly as colorful as this one right here, you can see on your screen. Pick 3 or 4 numbers and tell me what type of melanin they have, eu or pheo, and also the amount of the melanin being produced. A little bit, and a lot. All right, so something along those lines. Record those off to your side. I'm going to have you share those with me a little bit later on. Please pause this video to do that. Now we get to talk about the Goosebump muscle, which is the Arector Pili muscle. As you can see in this picture here, it attaches to the hair follicle deep down in the skin, and when it contracts, our hair stands on end, we get goosebumps. It is smooth muscle, so we cannot control how this muscle behaves. It's involuntary. You'll have goosebumps, or this muscle will contract, sometimes in moments of fear or panic. Some people get goosebumps. Obviously, when we're cold, it contracts. But all those impulses are sent from the brain to this muscle to take care of something. And as we can see looking at this image, the Arector Pili muscle is located in the dermis layer of the integument. Here we can see a real cross-section of the Arector Pili muscle. You can see some of the loose connective tissue found within this layer of the integument. You do have this on your coloring sheet. I think it's F6. And there should be two different Arector Pili muscles to color. One of them is not labeled. Make sure you get both of those labeled and colored. Pause this video to take care of that. The last little bit in this video that we're going to talk about is our finger and toenails. We looked at this image a little bit previously. We talked about this red region underneath the nail. Those are epidermal cells. They are alive. Those cells make up our nail bed. We can see deeper down here, this is a different shade of red than up here. This solid, nice red color makes up what is called the lanula. We can see this through the top of our fingernail as a white crescent-shaped region at the base. The nail itself, that's the nail plate. So how do nails grow? Very similarly to hair, these epidermal cells, which make up the nail bed and the lanula, they are alive. They go through mitosis. They push upper layers upwards, and as the layers get pushed up and up and up, they keratinize and they die, and they actually become part of the fingernail. So our fingernail is made up of old epidermal cells. The lanula, right here, goes through mitosis at an increased rate. And because this goes through mitosis at an increased rate, more cells die quickly. That is why the nail goes outward when it grows versus upward. We can also see that the nail is thicker right in this region. That's because these cells, they go through mitosis as well, so they contribute cells to the thickness of the nail plate as well. So this concludes our discussion on the first part of the accessory structures, hair and nails. The second video that you're going to take a look at will cover glands.